Embryology, from zygote to implantation. The oocyte is fertilised in the ampulla of the uterine tube. The male and female pronuclei merge and the secondary polar body is ejected from the ootid. At this point it is called a zygote. The starting point of this video is the formation of the zygote. All of the timings shown in this video are approximate, as the timing of events will vary between individuals and pregnancies. At around 16 to 20 hours after fertilisation, the zygote has formed. By removing the anterior portion of the zona pellucida, the internal events of the zygote can be shown. The zygote is transported along the fallopian tube, also known as the uterine tube, in tubal fluid, and is assisted along by muscular contractions and movement of the cilia lining the tube. In circumstances where the cilia are unable to function, the zygote will still move from the ampulla through the isthmus to the uterus. Research has shown that DNA from the secondary polar body is present within the blastomeres at the blastocyst stage and may be involved in early polarization of the zygote. Interactions between the secondary polar body and the zygote reduce after the two-cell stage. At around 30 hours after fertilization, the zygote becomes a two-cell zygote. During cleavage, the cytoplasm is divided at each cell division, leading to the daughter cells being half the volume, meaning the zygote, encased within the zona pellucida, does not increase in overall size. The individual cells are called blastomeres. Around 2 days after fertilization, the zygote becomes a four-cell zygote. Cleavage is asynchronous, meaning that by the two-cell stage there is no synchronization of cell division. So instead of the cell count proceeding from 2 to 4, from 4 to 8, and from 8 to 16, etc., it is more likely to initially proceed from 2 to 3, and then from 3 to 4, etc. Around 2.5 days after fertilization, the zygote becomes an 8-cell zygote. At around the 8-cell stage, the blastomeres begin to change shape and tightly align themselves against each other, and by the time there are 16 cells, the zygote becomes a compact ball called a morula. This change is called compaction and is mediated by the concentration of calcium-activated cell adhesion molecules such as E-cadherin, along with the contraction of the cytoskeleton, which allows the cell to squash together more tightly. At around 3 days after fertilization, the zygote becomes a 16-cell morula. Up until this point, the expression of the OCT4 gene has led to the production of the OCT4 protein, which is believed to play a regulatory role in keeping the cells in their undifferentiated states. The top 4 cells of the morula have been removed from the model, so the inner cells from this stage forward will be visible. At around 3.5 days after fertilization, the morula becomes a 32-cell morula. At around 4 days after fertilization, the morula becomes a blastocyst. Around the time the morula enters the uterus, it starts absorbing fluid through the zona pellucida, and the outer blastomeres transport sodium and water into the spaces between the inner blastomeres. This process is called cavitation and results in a fluid-filled space called a blastocele within what is now called a blastocyst. The uterine endometrium secretes uterine milk, which provides nutrition to the blastocyst, which is absorbed through diffusion. The outer polarized cells have now specialized to become the trophoblast and express the homeobox transcription factor CDX2, which represses the molecules OCT4 and nanog that stop them from differentiating into inner cell mass. As expression of CDX2 requires the transcription factor TEAD4 and cofactor YAP, the inner cell mass is unable to express CDX2 because the Hippo pathway is activated, which prevents YAP from entering the nucleus. The inner cell mass expresses molecules such as OCT4, nanog, and SOX2, which are associated with developmental flexibility. The trophoblast does not express such molecules. The embryoblast end of the blastocyst is called the embryonic pole, and the opposite end is called the abembryonic pole. At around 5 days after fertilization, the inner cell mass settles on one side of the blastocyst. The first cells that form the inner cell mass express nanog, which perpetuates their pluripotency. They also express fibroblast growth factor 4, FGF4, which influences later cells to express GATA6. The cells are initially mixed together, then the GATA6 expressing cells move to the inner surface and become the hypoblast, while the nanog expressing cells become the epiblast. Any GATA6 expressing cells that don't make it to the surface undergo apoptosis. The zona pellucida is gradually eroded by uterine fluid, becoming thinner and finally breaking down around day 5, at which point the blastocyst hatches. The outer surface of the zona pellucida stops the zygote or blastocyst from adhering to the uterine tube or the uterus, and so implantation is delayed until after hatching. At around 6 days after fertilization, the epiblast and hypoblast cells align themselves to form 2 layers. The epiblast layer will later become the dorsal side or back of the embryo, and the hypoblast layer will later become the ventral side or front of the embryo. Some of the hypoblast cells differentiate into anterior visceral endoderm, AVE cells, at what will become the cranial end of the embryo. So together with the epiblast and hypoblast, the dorsoventral and cranial caudal axes are already determined at this stage of development. This is achieved by secretion of the signaling molecules lefty1 and Cerberus1, which inhibit the activity of the signaling molecules nodal and WNT in the epiblast adjacent to the AVE. This signal inhibition results in the primitive streak only developing at the caudal end of the embryo. Later, the AVE will initiate head induction by inhibiting bone morphogenic protein 4, BMP4, signals through the antagonist noggin and inhibiting the WNT signal through the antagonist DICOP1, DKK1, resulting in the primitive streak formation being restricted to the caudal end of the epiblast. The AVE also participates in the formation of the precaudal plate. The wall of the uterus is made up of layers. The lowest layer is the myometrium, which is a thick muscle layer. Above that is the lamina propria, or stroma, of the endometrium. This is divided between the basal layer and the functional layer, topped by the luminal epithelium consisting of tall columnar-shaped cells. The functional layer is further divided into an upper layer called the stratum compactum and a lower layer called the stratum spongiosum. It is the majority of the functional layer which is lost during menstruation. Blood is supplied to the myometrium through the arcuate artery. This has radial branches into the basal layer of the endometrium, where straight arteries supply the basal layer and spiral arteries supply the functional layer. At around 6 days after fertilization, implantation or nidation starts in the uterine wall. At this point, the blastocyst expresses adhesion molecules such as L-selectins at the embryonic pole, which interact with L-selectin ligands expressed by small projections from the endometrium called pinapodes. The production of the anti-adhesive mucin 1, MUC1, is upregulated during the luteal phase and remains high during the receptive period and stops the blastocyst from attaching to unsuitable locations, and it is downregulated around the area of attachment. Other adhesion molecules are also necessary during this stage, including integrins. One of these is the heterodimer alpha-5 beta-3, which is part of the endometrial recognition process and is expressed by both the trophoblast cells and the endometrium, although only the endometrium expresses its ligand. Another adhesion molecule, called trophinin, is also expressed by both the trophoblast and the endometrium, and is also believed to trigger the cytotrophoblast to differentiate into the syncytiotrophoblast by signal transduction pathways. The syncytiotrophoblast is a multinucleated mass with no discernible cell boundaries, and also produces human chorionic gonadotrophin, HCG, which is transferred to the maternal blood supply via lacunae in the syncytiotrophoblast and maintains the production of estrogen and progesterone by the corpus luteum. By the end of the second week, enough HCG is produced by the syncytiotrophoblast to give a positive pregnancy test. The production of HCG increases until the 8th week. It then declines. However, by the end of the first trimester, or 12th week, the placenta produces enough progesterone and estrogen to maintain the pregnancy. At around 7 days after fertilization, the syncytiotrophoblast expands and starts moving between the cells of the endometrium, spreads out and expresses lytic enzymes such as matrix metalloproteinases, MMPs, which begins breaking down the endometrium. The spreading syncytiotrophoblast also induces apoptosis in the endometrial cells, making more room for the blastocyst to expand into. At this stage, the eroded maternal tissues also provide nourishment for the blastocyst. Once the luminal epithelium has been breached by the syncytiotrophoblast, the process of decidualization begins, at first locally, then eventually spreading to occupy most of the endometrium. This process causes the stromal endometrial cells to accumulate glycogens and lipids and lose their elongated shape, becoming polygonal. At the same time as the decidual reaction takes place, the leukocytes within the endometrial stroma secrete interleukin-2, which prevents maternal recognition of the embryo as a foreign body. After decidualization, the different areas of stroma can be identified as the decidua basalis, which is the area between the embryo and the myometrium, the decidua capsularis, which is the area between the embryo and the luminal epithelium, and the decidua parietalis, which is the rest of the luminal epithelium within the uterus. An amniotic cavity now forms between the epiblast and the cytotrophoblast. At around 8 days after fertilization, the exocellular membrane forms from the hypoblast. This membrane is also known as Husser's membrane and is a short-lived structure. The cavity beneath the exocellular membrane is the exocellular cavity and is also known as the primitive yolk sac. At around 9 days after fertilization, the blastocyst continues to sink into the uterine wall an extraembryonic mesoderm forms. The syncytiotrophoblast continues to grow larger, and the exocoelomic cavity is now called the primary umbilical vesicle. At around 10 days after fertilization, the extraembryonic mesoderm widens and the extraembryonic coelomic spaces appear within it. The syncytiotrophoblast continues to grow and the lacunae within it start joining together to form lacunar networks filled with maternal blood. At around 11 days after fertilization, the blastocyst becomes deeply embedded in the uterine wall and a closing plug of fibrin coagulum forms over the top. This will eventually be sealed by migrating uterine epithelial cells. Apart from at the position of the connecting stalk, the extraembryonic mesoderm completes dividing into the extraembryonic splanchnic mesoderm adjacent to the primary umbilical vesicle and into the extraembryonic somatic mesoderm adjacent to the cytotrophoblast. At around 12 days after fertilization, the extraembryonic coelom grows larger. The primary chorionic villi start to form in the cytotrophoblast, and the connecting stalk starts to migrate around the extraembryonic splanchnic mesoderm to a position opposite the anterior visceral endoderm. All of the abbreviations used in this video are shown on this page.